Okay, ladies and gents, today we're going to be talking about one of the most important macromolecules in our physiological functions in biology. So any type of cell needs these in order to carry out any function. If we didn't have this macromolecule, nothing would work in our bodies. And this is a product, this is information that's coded in our DNA. And I'm going to talk about kind of how we make proteins, how we use them to improve our body's functions, and what their actual purpose is inside of different organisms. Okay, so I'm going to go into hiding. And then we are going to address proteins. So if you look up here, proteins, the making of. So it sounds like a short that we're talking about. We made a movie or something. But essentially what we're going to be addressing is how proteins are actually formed inside of our bodies. So we've got proteins. We've talked about what they are. They are one of our big four macromolecules. So our big four are carbohydrates, lipids, proteins, and nucleic acids. So proteins are a macromolecule. So as a macromolecule, they are going to have a monomer. All right, and they're going to have another name for the macromolecule called polymer. So in our proteins, the monomer is going to be your amino acid. You put a bunch of those together, and you form what we call a polypeptide. Okay, so polypeptide and protein, they mean the same thing. Proteins, they usually have this other connotation where they're folded together, and I'll get to that in a second. But we have our amino acids that are put together to form a polypeptide. So to make this polypeptide, we do some kind of bonding, right? You've got to bond these monomers together. And this is type of bonding that we deal with amino acids. We call this peptide bonding. All right, you want your peptides to be put together so you can actually make your polypeptide or your protein. Okay, so to get to this step, right, to actually make our proteins that we use to carry out our different functions, right, each protein is specific. And the blueprint for building these unique proteins that are used for various different functions is found in our DNA. Okay, so our DNA is actually storing this it stores information in the form of, or stores your genetic information for later use. And so that blueprint gets read, and then we're able to make proteins based on that blueprint. Okay, so the DNA actually has the chain or the order in which we put our amino acids together. So we're not going to go into today how or what is read, right? I just want you to have a broad overview because we're really concerned about the function of proteins. Okay, so to at least make these, all right, we have to go back to the central dogma of biology. And the idea that you have your information in your DNA, and when you have that information in your DNA, it's going to be transcribed into RNA, right? You transcribe it. Transcription means you're just taking it from one form into another, right? And then when we go from RNA, we translate it into our protein, right? Protein is a totally different language. This language is made up of nucleic acids, right? And this language is made up of amino acids. So it's like where you go from, initially I give you a paragraph, I ask you to summarize it. That's pretty much when you transcribe it. But then if I say take that paragraph you made shorter, you summarized, and then translate it into French, same thing, right? You're changing the total language in which we're dealing with it. Okay, so whatever our DNA says is going to tell us whatever our protein is going to be. Okay, but let's talk about how proteins are made. So with our proteins, we get our chains. So you get your chain of amino acids, all right, and they have all different properties. Then we have different R groups. So we got different properties. These are called R groups. So if you look at the actual structure of an amino acid, right, you've got a nitrogen. Remember, proteins have carbon, hydrogen, oxygen, and nitrogen in them. That makes them unique. We have carbon. Those carbons are double bonded to this group right here, which has an OH. Okay, And then we've got here our R group. So our R group, there's 20 different R groups that are present in nature, at least that we use. And so based on this amino acid, right, these things get put together. And based on their R group, this gives it its unique properties. So whether or not it's polar, non-polar, whether or not it's got sulfur in it, which is going to make extra bonds, all different things. Okay, so proteins, different R groups, you've got these chains. What these chains do is the chains will fold together. So when they fold together like that, they're going to generate different forms, right? So if we take a string and you wrap it up, right, it's going to look very different. If you take a string and say start knitting it together, you can make something like a hat or a scarf. Okay, that whole thing is unique to that particular setup, right? So the chains fold together, they make this secondary structure. We got tertiary structure when those different folds interact with each other, and we have quaternary structure. Quaternary. I cannot spell today. I'm pretty sure I spelled that very wrong, but you guys understand what I'm getting at. So quaternary, it's like fourth level, and this is where we actually put two proteins together, two different chains together of amino acids. And a lot of our proteins are actually made this way. One of your best examples you hear about a lot is hemoglobin. And that's actually made up of 4 different protein units that are put together. So yes, they get folded together. That ends up leading to our function. So remember when I said structure leads to function? Based on how they are folded together, it's going to influence their function overall. So whether or not they're going to be a building block protein like our cytoskeleton, or if they're going to be something like an enzyme. Okay, and that's what we're going to be addressing today, is this idea of an enzyme. So with proteins, a lot of our proteins are enzymes, okay, but not all enzymes are proteins, right? Not all catalysts are proteins, okay, but let's look at what these actually allow us to do. Okay, so if we think about enzymes, right, these are proteins, okay, and an enzyme will function as a catalyst. So these are proteins that function as a catalyst. So let's review what a catalyst is. So a catalyst is something that speeds up a reaction. Okay, so you can think of it like putting lighter fluid on a fire, right, to light the wood. That's going to speed up your combustion reaction. Inside of our body, for a lot of the stuff that we need to actually have occur, the chemistry in it, we have to use these catalysts or these enzymes to actually make it happen. Because if we don't have our enzymes to do that, they're not going to actually ever occur, right? The activation energy is going to be way too high for it actually to happen. Okay, so proteins function as enzymes. Okay, so enzymes, they're catalysts, they speed up reactions. So each enzyme only carries out one part of a reaction. Okay, so you guys have probably heard the idea of a lock and key. So the idea that if we have our protein, so let's say I draw my protein like this, right? This is my enzyme. Okay, so based on my enzyme, all right, that's gonna have this thing we call an activation site. And based on the activation site's shape, okay, that's going to influence whether or not it's able to interact with something. So if I have a bunch of substances that are available, we'll call these substrates. Substrates are things that interact with enzymes. So if I have 3 of these substrates, if we look at our activation site and the shape of it, we can see that of these to make our enzyme work, this one's not going to work because it doesn't fit the shape. This one won't work, but this one will. Right? The circle one will. Right? That's going to actually be able to fit that. So if our substrate is able to get into our reaction site, we can then speed up our reaction. So if we say take a common reaction, some kind of carbon, so some kind of carbon compound like ethanol or something like that, and we react it with oxygen, gas, right, we're going to be able to get fire, right? We're going to get energy released. Okay, and we're going to get carbon dioxide and water outside of the other side of it, right? This is our common combustion reaction. You got some kind of carbon compound reacting with oxygen to make carbon dioxide and water. Okay, so if we say we want this reaction to occur, if we've got our pieces of wood, right, that are here in our, like board during the summer, getting ready for the winter because we're out in the wilderness, right? You've got your logs that are sitting out there in your pile, waiting to be burned, right? These logs right here, they're on fire, right? If you look at Brown County Park, it's on fire. There's a forest fire right now because this reaction is spontaneous, right? It will happen. There's like 1 or 2 molecules out of every trillion of them that is breaking down through this particular reaction. Okay, it's occurring. Okay, but is it occurring very quickly? All right, and that's because if we think about the energy of combustion, right, you've got to get past this energy to make it happen. Right, you actually have to input energy into the reaction to then get it to go, to keep going. Okay, so our logs are on fire, right, but very slowly. But let's say we want to burn one of those logs. We have to input some kind of catalyst that's going to speed it up, that's going to change that energy. So we've got our reaction, right? If we add our normal curve, right, where we've got our normal amount of energy it takes to so this is the amount of energy it takes, say, naturally. We want to take that energy and we want to make it so that it's easier to make it happen. So if we add a catalyst, so say an enzyme or something that allows for combustion, that would be scary. Like something that's going on inside of your mitochondria, your little combustion cells. We're going to change that to where our catalyst and our energy is going to lower the relative amount. All right, so we call this our activation energy. Right, and enzymes in our body actually lower activation energy. Right, they make it easier for reactions to occur. Okay. All right, so that's a quick tutorial on our proteins. Okay, so quick tutorial on proteins. So that proteins are one of our macromolecules. They're made of amino acids. They get bound together to make a polypeptide. Those things fold together. It leads to the final structure of the protein. Right? It comes from our DNA. That's how we get our final structure, is based on the sequence of amino acids we receive, based on what our DNA says. Okay, those things get folded together again. And then the structure of our enzyme leads to the function. So based on how it's put together, it's going to cause different things to react. So our proteins that function together, they work as a catalyst, or as enzymes, many of them, not all of them, but some of them do. And those enzymes help carry out reactions. So we've got our lock and key, and so based on the substrate, we're going to get a reaction. If the substrate doesn't fit, it's not going to happen. We're not going to get any increase in our activation energy. Okay, so for reactions, if they require extra energy to occur, enzymes make that energy amount go down or get rid of it altogether and force the reaction to occur. All right, and this causes our enzymes to make our bodies function. So without enzymes, we wouldn't have a lot of our body functions. Things would just not work, right? These proteins are magical in that they make things happen. Okay. So again, watch this stuff, review it, do your cues, write your summary, and take your quiz. All right. Enjoy.